nine-cis-epoxycarotenoid dioxygenase. Nine-cis-epoxycarotenoid dioxygenase, nine-cis-epoxycarotenoid dioxygenase, NCED, at NCED3. NCED is an enzyme in the biosynthesis of abscisic acid ABA with systematic name 9-cis-epoxycarotenoid 1112 dioxygenase. This enzyme catalyzes the following chemical reaction. 1. A 9-cis-epoxycarotenoid plus O2 2-cis-4 transdantoxin plus a 12 apo-carotenal 2 9-cis-phylloxanthin plus O2 2-cis-4 transdantoxin plus 3 S5R6 56 epoxy 3 hydroxy 56 dihydro 12 apo-beta-carotene 12 Al3 9-cis-neoxanthin plus O2 2-cis-4 transdantoxin plus 3 S5R6R 56 dihydroxy 67 dihydro 56 dihydro 12 apo-beta-carotene 12 Al. 9-cis-epoxycarotenoid dioxygenase contains iron Roman 2. Genomic sequence Chromosome 3, NC 3074.8, 4,831,286, 4,834,015 complement. Gene family. NCED belongs to a gene family called carotenoid cleavage dioxygenases (CCD), which contains both CCD genes and NCED genes. CCD is available in all plants including algae, while NCED is currently only observed in land plants. Please note some algae CCD genes have been incorrectly named NCED. There are usually multiple copies of NCED in a species. Gene function. The enzyme catalyzes the rate-limiting step of ABA biosynthesis. Interestingly, though ABA is also produced in algae, NCED is currently only observed in land plants, suggesting ABA is produced in a different pathway in algae compared to land plant. Though first identified in maize/corn, it is now quite extensively studied in the model plant Arabidopsis thaliana Arabidops. The most studied NCED in Arabidopsis is the NCED3. Overexpression of the NCED3 gene improves the tolerance of transgenic plants to dehydration stress as well as to salinity stress. Overexpression also leads to the overexpression of other genes induced by drought stress. Transgenics containing NCED3 had greater root biomass, bigger pith size, and higher level of photosynthesis. It has been suggested that the NCED3 gene promoter contains G-box-like cis-acting elements which are responsible for dehydration-induced expression. Gene expression differs by plant organ. The gene may have a dual role in roots by either promoting or inhibiting the development of lateral roots. NCED3 functions in seeds by regulating the seed establishment and abortion maturation of the embryo and seed dormancy. Maternal ABA functions in the early stage of zygote development, while embryonal NCED3 expresses later for ABA synthesis in case of dormancy. Expression of the gene mainly happens in the maternal tissues in the basal part of seeds or funiculus. NCED3 gene expression responds to drought stress and salt stress. The signal caused by low moisture in the air is first induced by the stomata of leaves and transferred to other cells and tissues, which upregulate the expression of the NCED3 gene and ABA synthesis. Stomata closure limits various processes including air exchange, water loss, and outward release. The NCED3 gene is active and expressed under these circumstances. Accumulation of NCED3 mRNA and NCED3 protein was first found in the vascular parenchyma cells under drought stress, which suggested that plant drought tolerance relates to the development of plant vascular tissue. Also, overexpression of NCED3 gene can improve plant salt tolerance. Other NCED genes in Arabidopsis are less characterized. However, Tanner al. 2003 observed that other NCED is important in plants and seed development. For example, NCED12 and NCED9 genes are important in flower development, while NCED6 is important in seed dormancy and development. NCED5 is found to be important in seed dormancy, and it can interact with NCED3 for drought response.